Hello everyone, I am Anshu Gupta, and today I am going to present about my work on DP-HLS, which is a high-level synthesis framework for accelerating dynamic programming algorithms in bioinformatics. So first, I would like to start with a brief introduction to dynamic programming and its usage in bioinformatics. So DNA, RNA, and protein sequences are core biological molecules for life. They contain a series of 4 nucleotide bases in case of DNA, that is AGCT, in case of RNA, that is AGCU, or 20 different amino acids in case of proteins. And these are represented as a linear sequence of symbols. What's important is that the order of these symbols encodes biological information, which eventually determines the functionality of the organism. Now, since the last few decades, there has been a massive increase in the availability of all kinds of biological sequences, like from DNA to RNA to proteins, because sequencing cost has been decreased. We have now millions of sequencing data available in these databases. As you can see in this plot, numbers are shown in the log axis, y-axis, and this shows how bioinformatics has become a large-scale computing problem. Now, we usually compare these sequences to find how well different biological entities are related to each other. It can also look something like this in the form of trees, as you can see here, where S1 and S2 are more related to S3 and S4, where different S's are BCD sequences. We can find the sequence comparison in applications such as human ancestry to study different hominoid ancestors, how they migrated, or to study the evolution of different species on Earth, like how it is shown here for different vertebrates. Or in homology detection-based applications to detect genetic similarities between two species. For example, mouse and human, which is shown here. The chromosomes are labeled here and the color corresponds to the similarities between the chromosomes of two species. Now, it also finds its application in genome assembly, where we take a saliva sample, feed it into the sequencing machine, which outputs small reads, and then we assemble these reads to form whole genome sequences. Now, although I have mentioned the comparison of real biological sequences in bioinformatics, we also often deal with non-biological sequences, such as aligning electrical signals. We have added one such example for this over here. There's the sequencing machine, which sequences DNA and generates electrical signals, which gets converted to AGCTs. Now, sometimes this also involves aligning two electrical signals. So there can be various types of input sequences involved in alignment. It's not just aligning DNA and protein sequences. And this kind of problem gets featured in base calling applications. Now, for comparing biological sequences, we perform sequence alignment. Now, sequence alignment can be considered as a string matching problem, which tells us how well the given sequences match. Now, the eventual goal is to maximize the matches, minimize the mismatches, and inserts the gap wherever needed to maximize the alignment score. Now, it's not just about alignment of two sequences. We can also align multiple sequences together, which we call it as multiple sequence alignment. Now, a good example of the usage of multiple sequence alignment at scale is AlphaFold, which performs protein structure prediction and had also won Nobel Prize for its profound impact on biology and drug discovery. Now, to perform structure prediction, AlphaFold relies on multiple sequence alignment as an input, which is highlighted here. Now, let's understand the algorithmic paradigm behind sequence alignment, which is 2D dynamic programming, or we say 2D DP. Now, for example, if we want to compare these two input sequences, we'll first construct and initialize a 2D DP matrix as a first step. Then at the next scoring step, we will fill the cells where each cell value represents the best alignment score up to that point. And then at the last step, we perform the traceback to find the optimal alignment path. And hence, we get the alignment of these two sequences using dynamic programming. Now, the example that I mentioned right now, it's called Needleman-Wunsch algorithm. Now, Needleman-Wunsch introduced us as a general algorithm, but it has found its application in bioinformatics for performing global alignments, such as aligning two sequences end to end. Now, this dynamic programming algorithm is well known since decades, and it can be easily accelerated in hardware using 1D systolic arrays, which is proven to be quite efficient. But the issue is that the dynamic programming comes with a lot of variations, and I'll talk about some of these variations in detail right now. For example, the same Needleman-Wunsch algorithm can be repurposed in many ways. For example, we can modify the initializing scores like this. We can also modify the scoring functions, and we can also modify the traceback starting and ending points. And that's how we get something, what we call it as local alignment, which is used in the applications where we want to find local patterns between the sequences. Now, similarly, we can also extend these variations in other applications, such as performing semi-global alignment or performing overlap alignment. Now, all we need to do is that modify the initialization scores, scoring functions, and traceback points. So they all follow similar 2D DP paradigm, but support different applications. Now, it's not just about these variations. We can also see variations in the form of sequence alphabets. So far, I've shown you the alignment of DNA and protein sequences so that we can align two DNA sequences and we can align also two protein sequences. But it's not just about aligning DNA and protein sequences. We can also align raw signals, which we see in base calling. And also we can align two profiles as sequence alignment. Now, so far I've shown you variations which are mostly limited to some parametric configurations. However, I also want to emphasize that the variations can go beyond that. For example, we can also modify the entire scoring function to something like this if you want to align sequences using affine gap penalty, which means penalizing different gap lengths differently. Or to something like this if you want to implement Viterbi algorithm. Or it can be modified like this if you want to perform dynamic time warp. The emphasis here is that the variations also involves rewriting the entire scoring function itself or customizing your own scoring function. So here I'm showing a list of 7 different types of 2D DP kernels, which follows a similar 2D DP paradigm, but with slight variations, which are categorized here. All these kernels are being used by multiple highly cited tools and in many different applications, which are also listed here. But this is a very small subset of biologically relevant 2D DP kernels, which are possible with these variations. And there can be many more such kernels across bioinformatics using the same paradigm which we target in this tool. So now we got to know about the variations dynamic programming now. Now I'll explain about the various existing approaches to map the dynamic programming on FPGAs. So one of the such approaches using custom RTL-based hardware acceleration. So while the hardware accelerators of dynamic programming are well understood, all of them map to a specific type of kernel. For example, FPGA SW accelerates only Smith-Waterman kernel, Darwin accelerates Smith-Waterman with tiling, Darwin WG accelerates X-drop with tiling, and so on. So basically, they are all accelerating specific type of dynamic programming algorithms, and they are not often easily customizable to support new applications. And since these implementations on FPGAs are usually defined by low-level hardware descriptive languages like Verilog, it typically requires to redesign things from scratch to support or extend it to new applications, which takes a lot of implementation time. Another option that people have used to leverage the programmability on FPGAs is using high-level synthesis, or in short, HLS. Now, HLS enables user to write their code in high-level languages, which gets converted to Verilog code, and thus it improves the design productivity. However, in our experience, we have seen that it requires months of effort to design anything custom using HLS. It requires user to know the right usage of pragmas or having certain expertise in the hardware design to match the custom RT level performance. Now, there's another approach that is software programmable accelerators. But the key idea here is that to use a fixed hardware architecture to improve the performance, but add software-related programmability on top of that to keep the flexibility. So the one such example for this type of accelerator is GenDP, which is also one of the DP-based frameworks targeting bioinformatics applications. Now, the advantage of GenDP is that it uses a fixed hardware with flexible PE interconnects and provides their own ISA for programmability and flexibility to support new kernels. However, custom ISAs typically creates overheads from extra steps such as instruction fetch and decode stages on FPGAs. Now, GenDP might be good for ASIC, but from FPGA point of view, GenDP loses performance over programmability. Hence, the main issue has been breaking the programmability and efficiency trade-off. Both HLS and software programmable accelerators provide high flexibility to cater to a set of applications, but at the cost of efficiency. So the ideal accelerator should provide algorithmic flexibility as well as near custom RTL efficiency. Hence, our main objective of this work is to develop a framework which is 1, highly programmable, where user can define their own custom kernel. 2, it is quite efficient on FPGAs, which almost give near RTL performance. 3, it should be accessible to non-hardware experts as well. Now, keeping these 3 objectives in mind, we introduced a new framework called DP-HLS. So the main insight behind DP-HLS is that a broad class of dynamic programming algorithms follow the same hardware primitive, that is linear systolic array architectures. So we created a new layer of abstraction on top of HLS, leveraging this insight, which separates the front end and the back end, where front end gives you the programmability and back end gives you the flexibility. So the unique thing about DP-HLS is that in the front end design, in addition to parametric configurations, it also allows user to add their own custom function. And for that, users are not required to have any hardware design expertise. Now, in the back end, DP-HLS uses certain HLS directives, which enables it to generate efficient linear systolic architectures. Each of these architectures customized with its own unique functionality, operations, and data flow patterns to match the specific needs of the target algorithm. Now I'll go through the steps of providing the customizability in the DP-HLS frontend. First, user can specify their own initialization scores for rows and columns of the DP matrix, since that varies with multiple applications. User can also customize the type of sequence alphabets, scoring parameters, and traceback starting and ending points. User can also modify the scoring function as well as they can modify the traceback strategy. User can also provide how many processing elements they want to be there in one systolic array architecture, or how many kernels they want it to be implemented and executed in parallel on one single FPGAs. And user can also customize and modify their own host code. Now I'll talk about the backend optimizations, which is hidden from the end users, as it encapsulates various HLS directives that do not require user modification. So DP-HLS performs refund parallelism in the backend. Score computations are performed in the chunk-wise fashion, which eventually generates 1D systolic array architectures. Now, all the local input and output buffers are partitioned for parallel access. And for the traceback logic as well, DP-HLS performs certain optimizations, such as memory address coalescing and independent access to memory banks by each processing elements to ensure parallelized execution. So now we know about the details of the DP-HLS. Now I'll talk about the key contributions of the DP-HLS and the results. So the first contribution is that the DP-HLS showed extensive versatility, which we demonstrated by implementing 15 different DP kernels, which all follows the similar paradigm. And DP-HLS is not just limited to 15, it can support all possible type of kernels, which follows the same 2D DP paradigm. Along with versatility, DP-HLS showed near custom RTL performance, thus overcoming the efficiency programmability trade-off compared to other approaches. And the results are shown in this figure, which shows that DP-HLS got similar throughput and resource utilization compared to custom RTL approaches. DP-HLS is about up to 41 times more cost efficient compared to CPU baselines. Now, cost efficiency is equal to actual throughput normalized by the instance cost. Compared to GPU baselines, it gives up to 17.7 times higher cost efficiency as shown here. And we also measured the raw throughput without the cost normalization, and DP-HLS was 9.5 times better. We also compared it with the latest NVIDIA Hopper GPU, which has DPX instructions, and it is 2.27 times more energy efficient. Hence, DP-HLS can be very useful in low power-based applications. DP-HLS has shown massive improvement in the design productivity, and the kernels can easily be deployable on AWS FPGA instances. Also, DP-HLS supports both short and long sequence alignments through tiling-based approaches. Now, DP-HLS artifacts are open sourced, functional, and reproducible. So for more details, you can also look up the GitHub repository. Now, I'd like to conclude my talk with some key takeaways and future applications. So as a key takeaway, I want to highlight that DP-HLS leverages the key insight that a broad class of 2D DP algorithm can be mapped to the same hardware primitive. And following this insight, it creates a new layer of abstraction in the HLS flow, which separates the front end and the back end, where the front end parts provides a software-like programming model to allow users to define their own 2D DP kernel without requiring any hardware design expertise. Whereas in the back end, it creates an efficient linear systolic array architecture, each of them optimized for the target algorithm. Now, at the end, it achieves a competitive performance compared to custom RTL designs on FPGAs. So in future, we can leverage the same insight that we found in DP-HLS for other sets of applications too, sharing the same hardware primitive. For example, in AI workloads, operations such as convolution, matrix multiplication, they all naturally map to single hardware primitive, which is 2D systolic arrays. And it also comes up with its own variation and customizations, like you have different precisions, different quantizations, sparsity, different types of nonlinear functions, etc., thus making them well suited for future extensions for DP-HLS type framework beyond bioinformatics. And before ending my talk, I would like to acknowledge my advisor, Yatish Surakia, and my co-authors, Inky and Jason, for the help and support. The work is performed in collaboration with AMD Omics Group. Our artifacts are open sourced. Kernels, which are generated by DP-HLS, are deployable on AWS FPGAs. We also created a wiki with all the details. So if you want to find more details about our work, please check out the wiki or the QR code for the GitHub repository. Thank you very much.